infections are one of the most common and often misunderstood health issues, especially among women. Caused by an overgrowth of a fungus called Candida, they can lead to itching, irritation, and discomfort. In this video, we'll take you inside the laboratory to show exactly how yeast is identified. You'll see the process that helps lab professionals detect and confirm the presence of yeast. Let's begin. For sample collection, a high vaginal swab is often obtained using a Köppen swab. This sterile swab is designed to preserve the integrity of the sample during transport to the laboratory, ensuring accurate results. A Köppen swab is a sterile, single-use collection device designed for clinical sampling. It features a soft tip that gently collects cells and microorganisms and is paired with a transport medium that keeps the sample stable until it reaches the laboratory. In high vaginal swab collection, the Köppen swab helps ensure an accurate, contamination-free sample for yeast identification. Once the high vaginal swab sample is collected, the next step in the laboratory is to culture it on Sabouraud dextrose agar, which is specifically designed to promote the growth of fungi, like yeast. Step-by-step process. 1. Label the Sabouraud agar plate with patient details and date. 2. Using sterile technique, gently streak the swab onto the surface of the agar. 3. Rotate the swab to ensure the sample is evenly distributed. 5. Incubate the plate at 25 to 37 degrees Celsius for 24 to 48 hours, depending on the lab protocol. 6. Observe the plate for colonies characteristic of yeast, which may appear creamy, smooth, and white to off-white. After incubating the Sabouraud agar plate for 24 to 48 hours, yeast colonies should begin to grow. These colonies are usually creamy, smooth, and white to off-white in appearance. This growth confirms the presence of yeast in the sample and allows laboratory staff to proceed with further identification and testing if needed. After yeast colonies have grown on the Sabouraud agar plate, the next step is to prepare a wet mount. This allows us to examine the yeast under the microscope for further identification. Step-by-step process. 1. Using a sterile loop or swab, carefully pick a small portion of a yeast colony from the agar plate. 2. Place it in a test tube containing water or saline. 3. Make sure enough colony are obtained. 4. Gently mix the colony with the liquid to create a thin suspension. 5. Add 1 drop of the suspension to a microscope slide. 6. Place a cover slip over the preparation, avoiding air bubbles. Observing the wet preparation under the microscope allows us to see the characteristic features of yeast. Under high power, yeast cells typically appear as oval or round budding cells. Some species may also produce pseudo-hyphae elongated cells connected end-to-end. These microscopic characteristics, combined with the culture results, help confirm the presence of yeast and guide further identification or testing. To know which Candida species is present, further testing is to be done. The most common yeast is called Candida albicans. Candida albicans, the most common cause of candidiasis.
Thanks for watching. Please like, share and subscribe. And remember to click the notification bell to know when future videos are released.